Hello, I'm Darren Griffin. I'm joining you from my labs here at the University of Kent, and it's a very special year this year because it marks the 30th anniversary of the world's first live births following pre-implantation genetic testing. That is the genetic diagnosis of an IVF embryo before it implants. Now, in April of 1990, a team led by Alan Handyside and Robert Winston announced their findings in the journal Nature, and this coincided with the birth of the Monday twins in July. They are, well, we need to wish them a very, very happy 30th birthday, albeit somewhat belatedly. So there's the headline, and there are the Monday twins. So it gives me the greatest of pleasure to be welcoming today the man who is widely considered to be the father of pre-implantation genetic testing, Professor Alan Handyside. Professore, hello.
Hello, Professor Griffin.
Or should I say hello, father?
I demand a paternity test.
Well, it's 30 years and you do not look a day older.
Thank you very much. I wish I could return the compliment.
You've been practicing those answers, haven't you? Right. It is 1990. We're casting our mind back to. It was quite a turning point in your career. But in point of fact, you've been publishing for about, what, 14, 15 years before that? Before 1990, what would you say were the highlights of your career?
Well, you're quite right. My research career began in 1974 as a PhD student, working with Martin Johnson in anatomy in Cambridge. And at that time, I developed an interest in early mouse embryology, and particularly how the two first cell lineages within the embryo develop in the first few days of development following fertilization. And those lineages are the outer trophectoderm that forms the blastocellular cavity and the inner cell mast cells from which the fetus is derived. And following my PhD research, I went on to do a postdoc and actually discovered that you can visualize that the cells polarize at compaction, and that the mechanism, part of the answer of the mechanism of how the cells allocate cells is that they become polarized. And so this led to the polarization hypothesis for the allocation of cell lineages in the mouse blastocyst. And I did that with Martin Johnson, of course, and Karol Ziomek, who went on to have a series of classic cell papers that used those markers that I developed to look into that in more detail. But subsequently, having been away in Johns Hopkins for a postdoc, and I came back to work at the MRC Experimental and Teratology Lab in South London and had my own lab. Working in collaboration with Martin Hooper, I derived one of the very first mouse embryonic stem cell lines, a cell line called E14 for Edinburgh 14. And I'm very proud to say that stem cell line is still widely used in stem cell research today, some easily 30, 35 years after it was derived. And I subsequently then led a team which demonstrated, was one of the first demonstrations that you could inject these embryonic stem cell lines into the mouse blastocyst. They would form part of the embryo and actually contribute to the germline. And we'd isolated cells that were deficient in HPRT, the enzyme hyperxanthine phosphoribosyltransferase transferase, in the hope that we could develop a mouse model of the human inherited condition, which results from deficiency in that enzyme, Lesch-Nyhan syndrome. And indeed, we were able to do that. And that was a very exciting period of my life. And actually, we ended up publishing back to back with Martin Evans, who I previously worked for, and who subsequently was awarded the Nobel Prize for gene targeting in embryonic stem cells.
Wow, there's an awful lot I didn't know there. So I think it was probably about 1989, 1990 that we first met. And now, if I recall correctly, we both discovered we're both left-handed, and we had a very obsessive way of making tea. Yes, likewise. So something to do with warming up the cup first, and then making sure it was, you pour the hot water right the way through the tea bag, and so on. And it was a time where really you were just on the cusp of all of this. And did you have any inkling at the time that you were onto something big?
Well, I think people forget that I was actually involved in animal-based research, particularly mouse embryo research, for about 5 years before we actually got to the first clinical application, which clearly was towards the end of 1989. So I'd been using the transgenic mouse that we developed, using the embryo stem cell technology and the injection into blastocysts as a mouse model. So I was able, working with Marilyn Monk, to actually use this and as a proof of principle, if you like. So around about 1986, when my contract with the MRC was coming to an end, I had to make a choice between going further with embryonic stem cells and getting into stem cell research, or becoming involved in human work. And in particular, there were various preprints circulating at that time indicating this possibility that it might be possible to diagnose inherited disease at a very early stage of development, so that you could then selectively transfer embryos that weren't affected. So I actually made a conscious decision and was proactive in approaching Robert Winston, now Professor Lord Robert Winston, about the possibility of working with him. And so it was a very conscious decision, because I realized that because of my background in mouse embryology, that I would be very well placed to help to make this a possibility. So I went actually to join Robert Winston at Hammersmith Hospital with two main aims: to develop methods for preimplantation genetic diagnosis, and also, if possible, to derive human embryonic stem cells. So I achieved one, not the other.
Excellent, excellent. So cast your mind back then to probably what must have been only the second or third time that we met. And that would have been about the time that the TV cameras were all over the Hammersmith. So the Mundy twins were probably just born. The Nature paper was just out. The Hammersmith was full of TV cameras. So suddenly you were a media star. So what are your memories of that time?
It was a tumultuous time. There's no doubt about that. And but for several years in the run-up to the first PGD application of PGD and the debate in the Houses of Parliament, I'd had regular visits from journalists and politicians and so on. And but it certainly took off after the publication. And I had journalists and film crews from many countries around the world come and visit. And of course, they all particularly wanted to film embryo biopsy because it was visually quite striking and iconic of the approach that we were taking. So it was very difficult with an expanding team of scientists and students. It was very difficult to get any work done. And so it was extremely stressful.
Well, there they are. And there they are on top right at a very early age. They weren't born on a Monday by any chance, were they? That would have just be too poetic.
Not that I can recall.
Now, it was, I remember the Mundy twins were actually the second case, but they were first case to be born. And actually 3 days later, we had some very bad news, I seem to recall, that one of the twins didn't make it. I don't remember their names.
Yes, well, actually, it was, I think this is where memory lets us down. Those twins are actually not the Monday twins.
No, exactly.
One of the twins in that pregnancy was actually stillborn, but there were no signs of any fetal abnormalities, and they were both female, showing that we've got the diagnosis correct, because both of these couples were at risk, as we'll come on to, of conditions that are caused by genetic defects on the female X chromosome.
Yeah, so if memory serves, they were the first case that you did, but the second birth, if you like. So I think you did the biopsy and diagnosis on them a few days before the Mondays, but the Mondays were just born a few days earlier. Like, as you say, the memories do tend to fade a little bit on this.
That's right. So the other set of twins, which were born in very close, at a very similar date in July of 1990, were actually Natalie and Danielle.
Yes, of course.
And from another patient. And they were at risk of a condition called adrenoleukodystrophy.
Mm-hmm. Now, one of the things that you've just alluded to a little bit, and people don't always realize, is that this largely coincided with a huge debate in the country in general, and that led to the Human Fertilization and Embryology Bill. Now, how do you remember the national mood at the time, and that very big free vote in Parliament?
Well, again, it was a very busy period. All of us involved in assisted reproduction at that stage were involved in providing evidence of benefit for embryo research, for example. But it was a time when people like Enoch Powell and the pro-life group were very vociferous. And actually, Enoch Powell brought forward a private bill, and it considered that any research that was done, even at a very early stage, was equivalent to abortion. So he took the view that life started at conception, at the point of fertilization, and so on. So I think the opinion was divided, and most importantly, it was very divided within Parliament. So as soon as I joined Robert Winston in late 1986, we were very much involved in publicity. And it was almost every day there were stories in the press and journalists wanting to talk to us. And so it was very difficult. But of course, I think where we did contribute was the fact that as we were developing preimplantation genetic diagnosis, people could understand that this was very powerful for couples at risk of really very serious conditions, and could see the benefits that this might deliver. And of course, at the end of the day, Maggie Thatcher actually went through the yes lobby and voted for embryo research.
What would we have done if she had said no? If the Parliament had said no?
That would have been interesting. I mean, I think we would have had very different careers. I think clinically, pre-implantation genetic diagnosis would probably have gone ahead because we'd already started it clinically. There may have been some delay because obviously the bill set up the Human Fertilization and Embryology Authority to regulate assisted reproduction in the UK, and they would have had to have taken a view. So I think PGD, as it was then known, or pre-implantation genetic testing, as we now know it, would still have gone ahead. But a lot of the other associated research might have been very difficult.
Absolutely. I mean, Yeah, I mean, it's just hard to imagine, really, just what a battle it would have been. And is it right that it was the first free vote in Parliament, or am I just misremembering that?
It was certainly a free vote.
Mhm.
So it was very difficult to say how it was going to go, and we didn't know until the very last moment, really. We didn't have an inkling. But in the end, it was passed with quite a large majority. And that was, I think, down to a huge effort that the scientific community, including my PhD supervisor, Martin Johnson, Peter Brodie, Dame Anne McLaren particularly, and others, and many others, in providing information to MPs about the potential benefits. And that, I think, in the end, swayed uh, the Houses of Parliament.
Yeah, I think it was something like 3-1 in the end, wasn't it? So convincing, but not unanimous.
No,
Yeah,
no.
absolutely. Now, you mentioned Robert Winston a few times. So what was your relationship with him, work-wise, and what is it now?
Well, I think really for me, Robert turned out to be the ideal clinical partner. If I'd had to choose anyone, he understood the importance of the work and was very excited. And he was very committed. And I think people don't give him full credit, perhaps, for the significant contribution he made. Using his media experience, he had in the 70s had various television programs and has always been a very powerful communicator. And that was enormously important. And he was prepared to go out there and spend a lot of his time promoting the idea that there could be benefits from human embryo research, that it would benefit in fertility treatment and so on. And so I was actually very happy. People often ask me, people said, well, look, it seems as though Robert Winston has done all of this. And we know that you're working in the lab to do the technical aspects. And indeed, I did all the original embryo biopsies, for example. But really, it worked very well. And I was very happy for Robert to be the front of house, as I used to say. He's much better at communicating with the public, whilst I worked backstage and kept the show on the road.
Are you still in touch?
We are indeed, actually, and I was very pleased to receive an email from him. So I recently was invited to guest edit an issue of the Reproduction journal, and in which I recorded a video, and I was very pleased to be back in contact with Robert, who enjoyed the video, and we shared various memories. I particularly, within the video, reminded him of a remark I made, because I was actually worried before we did the first cases. I knew from my animal background with mouse embryos that if you remove the zona pellucida, which is this glycoprotein coat that surrounds the embryo at early stages, and you replace that embryo in the uterus at an early stage when it only has 8 cells, which are only loosely held together, then in fact there were no pregnancies because the embryo was destroyed, presumably disaggregated within the uterus. And I had been making very large openings in the zona at that stage in order to remove a cell to do the diagnosis on the embryo. And he said, so there's a danger that the baby will fall out of the pram. And he was very pleased to be reminded of that.
Yes, it's a very Robert style comment. Now, think back to your days at the Hammersmith Hospital. I've got a picture that you might quite like to see. Remember this? So,
yes, I do.
And the little shed, that's Wormwood Scrubs behind you, if I recall correctly, and you actually camped out on that little shed that's to the left of our picture. Is that right?
Yes, indeed. And in fact, we weren't in the building that you can see, which is a brick-built building. My lab and our offices were in porta cabins on the other side of that building. And yes, indeed, we were within sight of wormwood scrubs, and in the summer, we could hear the prisoners communicating to each other from one window to the next. And so there you see my team. I have a little more color in my hair. For any of you that can't recognize me in that photograph, I'm second from the right. To the right of me is John Lesko, who is the postdoc from Mark Hughes's lab, and we'll talk about him later. He was a very talented postdoc in Mark's lab and did a lot of the original single-cell PCR work. Next to my left is Joe Connigan, who was one of the embryologists that did a PhD with me. And then one of the delights of my lab at that time was to have people from all over Europe. So next to Joe is Juan Tarin from Spain, who's now a professor in his own right. Then Elena Contagiani, who is now owns her own IVF clinic in Greece. And then Pierre Ray, who's now Professor Pierre Ray, who is has a lab in Grenoble in France.
So, and of course, Eleanor was a co-author on the Nature paper along with Kate Hardy, if I recall correctly.
She was indeed, yes, yes. So in fact, all the work at that stage was done by my various students and postdocs with my help, as it were. I did the biopsies and helped with the gel electrophoresis and the PCR.
You just can't help thinking, you know, you were making these world-beating discoveries at the time. Couldn't the Hammersmith Hospital done a little bit better than a Portakabin for you?
Well, I have to say that when I took the decision to join Roberts, I had an interview in his office, which was in a perfectly normal building, which is just beyond and closer to Wormwood Scrubs from the picture you can see there.
It's just to the right of the picture, isn't it, from there?
Left, I think.
Left, I think. Right.
But so he had an office on the first floor, and I naturally thought, well, you know, there's not a lot of space in this small building, but I probably offered a small lab downstairs, and, you know, let's see how it all works out. But then he led me out of that building, down the hospital corridor, and into some Portakabins. And I have to say, my heart sank when I saw the laboratory facilities and the offices, because these were literally single-story temporary buildings that were put up in the 60s. And of course, because they were sort of prefabricated construction, they were very hot in the summer and very cold in the winter. And so it was really very difficult. And there were very little. So I came from an MRC unit, which was very well equipped. I had all of the equipment I could possibly want. And so I was just lucky, for example, that I was able to inherit a lights micromanipulator, which I was able to adapt for embryo biopsy. But otherwise, things were very limited. So it was very much sealing wax and string in the good old British tradition.
Didn't you keep having your computer nicked from the Portakabin?
Well, that was, yes, I'm pleased you reminded me about that episode. That was really quite dramatic. So my office was on the far side towards Wormwood Scrubs, and there was a little passageway outside my window. And one morning I opened the door to see that two men had smashed my office window and were running off with my Macintosh computer, one of the very early ones, with the power supply trailing behind them, and never to be seen again. But of course, that was a time when computer memory was actually very valuable. So they would not have tried to resell the computer itself. They would have stripped it for its memory and sold the memory for, you know, on the black market.
Wow. Did you lose any data?
Yes, all my contacts, you know, the usual. Obviously, at that time, we didn't have mobile phones. So a lot of it was based on your computer and so on. So it was really quite traumatic. So we had to have bars put up on my office window and the labs. So, you know, those were the kind of conditions that I had to work.
Wasn't that just to keep you in?
Yes, something like that. Yes, yes.
Right. So think back to the approach that you used at the time in those first PGT cases. So you were amplifying a Y chromosomal repeat to diagnose sex. Now, with the benefit of hindsight, and with all the false positives and false negatives that can throw up, was it, with the benefit of hindsight, a good idea?
Well, I think you have to remember, it is easy with hindsight to say that it was a flawed approach. Obviously, we did publish our preparatory work, which is embarrassing now, but bear in mind that this was done with human embryos. And so, of course, that you're always very limited to access of material when you're working with human embryos. But nevertheless, we did do preliminary work that showed that it seemed to be accurate. But because this method was essentially set up to amplify a sequence, as you say, on the Y chromosome, the presence or absence of that amplified fragment really was the basis on which you said, well, if the fragment is present, it's a male embryo. If it's not present, it's a female embryo. But sadly, we understood really quite quickly that even with this sequence, which was repeated thousands of times on the Y chromosome, you did sometimes get amplification failure, which you couldn't detect. So it was very flawed from that point of view. And of course, we learned that very quickly because one of the 5 pregnancies we published in the paper in 1990 was subsequently, through prenatal diagnosis, shown to have been an error and was in fact a male. And so the couple who'd been advised to have prenatal diagnosis, because this was so early, so experimental, had to terminate the pregnancy, sadly.
And didn't a young PhD student quickly come up with a better method?
Well, let me see if I can remember the details. Yes, I think, all due credit to you working in Joy Delhanty's lab and others such as Santiago Monet. The development of fluorescent in-situ hybridization was certainly a turning point and was much better, particularly if you ran. So this is a technique in which you use DNA probes that are specific to particular chromosomes. You have to spread the nucleus from the cells that you remove from each embryo and then hybridize those probes to see whether that chromosome is present. And of course, the probes are labeled with fluorescent labels, and I think you did a lot of that work initially.
I did.
And of course, one of the challenges for you was to find a probe that we didn't have commercial probes, did we?
That's right, yeah.
So a lot of your initial work was actually to develop the probes that would allow us to do this.
Yeah.
And fortunately, of course, we had different fluorochromes, so you could use multicolor fish. And so eventually, we're able not just to have a probe for the Y chromosome, but also for the X, and a control autosomal probe, so that unlike the PCR reaction, we could indeed know that the fish had essentially worked. And of course, this turned out to be very accurate, even from single cells, and very quick. You can do this with a good probe in about 2 hours. I think you'd agree.
Yeah. I mean, that was, I remember it very clearly, actually. It was my 20-somethingth birthday, and finally getting the technology down to that window of 1 day, which was absolutely crucial to making it work. And I think one of my proudest things really is the fact that although you probably wouldn't use it now, you could use that as an approach for sexing even today. And we could spread the cells down a single cell and do the diagnosis that way if we were just looking for sex. And it's certainly not one of the things that's had, which we'll get onto later, all the debates of some of the other chromosomal diagnoses that we've done. I want to stay with sexing, actually, because as we saw from that headline that I showed a picture of from the Sunday Times, although our motivation was to be helping families that were at risk of transmitting sex-linked disorders, inevitably people were asking about social sexing. So I want a boy, I want a girl. What's your position on social sexing generally?
Well, I think, it was really the only thing we could contemplate, the only diagnosis we could contemplate at that time. And it was done in prenatal diagnosis also for X-linked diseases, was to identify the sex. But it was unfortunate that, of course, for thousands of years, parents have wanted to predetermine the sex of their children and their offspring. And I think that's what led very early on to the term designer baby. I'm rather frustrated. I can't put my finger on who was the first to coin that term, but it was very soon after we had done the initial work that the phrase came up. And then, of course, there was a debate about whether this should be allowed, whether parents, not just at risk of X-linked conditions, which of course typically only affect boys, but couples in general could have IVF, to predetermine or to control the sex of their next child. I personally have no problem with family balancing, but I would discourage patients from going abroad. So one of the first things that the Human Fertilization and Embryology Authority, that the bill that we've just discussed, set up, one of the first things they did was to have a public consultation. And they decided that actually the public was uneasy about that. And so they decided that that should not be allowed. And so in this country, and subsequently it became part of the law, we are not allowed to divulge in the course of making a diagnosis, unless, of course, it's an X-linked condition, or indeed there's an abnormality of the X chromosomes. But we're not allowed to identify that. But in my experience, I think patients need to think very carefully before they do this, because IVF and testing of embryos is not very easy. And I think patients often have unrealistic expectations. So you can have 5 embryos of one sex, for example, relatively commonly. And, so I think you need to think it through.
Yeah. That sounds like a competition, actually, for anybody who cares to watch this. Who came up with the phrase "designer babies" first? That would be interesting to see what the answers are and how far back it goes. It probably goes back a long way if we really look. But
if anyone knows, I'd be very interested.
Very much so. So yes, if you're listening to this, do email one of us with an answer. And if anybody can come up with the earliest, I'm sure we can find some sort of prize for you. So how about that? I thought about that top of my head just now. Right. So we went from sexing, and then the next obvious place to go was single gene disorders. And you've mentioned John Lesko already, but he was also working very closely with Mark Hughes. And there was quite a transatlantic collaboration going on there. And didn't Mark Hughes get sponsored by an airline or something? Or am I misremembering that?
Yes, I think he did. He was quite creative. He subsequently was sponsored by a star from American football.
Oh, really?
But that's another story. But yes, he was sponsored to the extent that I think it was Delta Airlines, but I may be wrong.
I think that's right, yeah.
I think it was Delta, basically paid for a couple to come from Houston, Texas, to the UK to have their embryos diagnosed in a case where they were at risk of cystic fibrosis. I think you have to think back to the early 90s. PCR was still in the very early stages. So when I started PCR, it literally involved moving rafts of tubes from one water bath to the next, from almost boiling temperatures in one water bath to the annealing temperature and so on for each cycle of PCR, the polymerase chain reaction. And reagents were very unreliable. And so given our very relatively primitive laboratory conditions and facilities, in fact, I met Mark Hughes, we'll come on to this, at one of the early International Society meetings. And he was based at Baylor College of Medicine in Houston, Texas. which is one of the foremost centers in the US and played a big role in human genome sequencing. And so it was clear that if we worked together, we could perhaps iron out some of the practicalities involved in this. And so we then were successful. So Pierre Ray working in my lab, and John Lesko working in Mark Hughes's lab, were able to develop what's called a nested PCR reaction, in which you amplify one fragment and then go on and amplify a second time a short fragment. And that within that initial fragment, and that was the secret to amplifying the region around mutations, which allowed us to pick up unique sequences, even at the single cell level.
Wow. And well, it obviously worked very well. And yeah, the idea of not only the nested PCR, but the heteroduplex formation to get the different diagnoses of affected, unaffected, and carrier, I thought was it was wonderfully elegant at the time, and you know, great credit to both groups for coming up with that.
Well, again, you know, it comes back to your remark about how little time we had. You've got to remember at this stage, we couldn't effectively culture embryos to the blastocyst stage. There were many people and groups that said, you know, we must culture embryos to the blastocyst stage, but I knew that clinically pregnancy rates were very low. So we really just had something like 12 hours to perform the analysis. So you're quite right. Because the main mutation causing cystic fibrosis, or the most common one, which I actually carry, 1 in 20 Caucasians carries this 3 base pair deletion. So out of the 3 billion base pairs, I'm missing 3 bases in the cystic fibrosis gene. And that turned out to be very useful because I could be endlessly bled for my lymphocytes, and we could do experiments at a single cell level without having to have affected embryos and so on.
What would the ethical committee say now, I wonder? But
well, yes, that's right.
yeah, I remember that 12-hour window very well, and I particularly remember one case where the patient completely forgot her appointment, and that would have been sort of okay, except that Robert was taking us all skiing the following morning. So the upshot was, if you recall correctly, we had to track her down, and instead of doing the biopsies in the morning, you had to do them in the evening. Leander, who used to spread the Leander Wilton, who used to spread the cells onto the slides, had to do that before midnight. And I had the job of basically working through the night to do the diagnosis, and basically picked up my skis and went to Switzerland just to send the facts through to Robert at the time. So yeah, I mean, they were very fraught days in many ways.
Yes, all credit to you. Yes, it was extremely stressful from that point of view. And I think, you know, it's remarkable what we're able to achieve, because let's remember, you know, in that first series of cases, we actually had 45% per embryo transfer positive pregnancy tests and 35% ongoing pregnancies. But I think in your particular case, and the one you've just mentioned, I think sadly all of the embryos in the end turned out to be males.
They were indeed, yes.
Mm-hmm. And therefore not available for transfer. But, you know, we nevertheless, you know, went the extra mile, yes, you might say.
And if I recall correctly, we were all in the air to Switzerland, and of course there was no means of contacting them. And apparently the patient literally went down on her knees and begged, put them in anyway. And I believe, and I could be very wrong about this, that the decision was taken to put in the 2 where we didn't get a diagnosis either way. So we were back to the 50% chance, but she didn't get pregnant.
Yes, I don't fully remember the details, but I know that in that situation, it was very frustrating to patients. So we had counseled them that would be a possibility.
Yeah.
Because it's a 50-50 chance. But even knowing that, it's very disappointing after all the process to get to that point. And
yeah, no.
So generally speaking, when they had calmed down and reflected, they decided that they were doing PGD to avoid the possibility of prenatal diagnosis, and that essentially they wouldn't take the risk.
Yes, yeah, very much so. And I think that normally that's ordinarily what would have happened. But I believe that most of the team were in the air on the way to Switzerland at the time. And yeah, I mean, you remember things differently, of course. Well, let's stay with chromosomes then, because we've largely done single gene disorders up until now. But running alongside that at the same time has been the idea of being able to diagnose chromosome disorders, partly because of the risk of Down syndrome associated with maternal age, partly with the possibility of reducing the occurrence of miscarriage, but controversially, the idea that it might improve IVF success rates, so-called, I think we called it PGS then, now PGTA. So we're still debating that right now, and I think you've got a bit bored of all this, haven't you? But for the purposes of the tape, as they say, give us your view.
Well, that would take at least an hour, if not longer, to deal with in detail. But I think the thing I would say is it's all credit to you again for the pioneering work, and Santiago Monet and other people that became involved, Yuri Velinsky, for example, as well, in Chicago in the United States. But those of us that were involved in basically researching the status of chromosomes in early embryos, particularly cleavage stage embryos, what came out of that as a legacy was really the awareness that there are indeed a lot of abnormal chromosome complements. So you'd normally have 23 pairs of chromosomes from the father and the mother that come together within the embryo to give you 46 chromosomes, 23 pairs, and so on. But actually, the early embryo has many abnormalities, some inherited, particularly in the egg, because during the formation of the egg, the wrong number of chromosomes is allocated to the egg cell. But these can also occur as the embryo is developing. And that was one of the big legacies of PCR. Then as this developed, of course, we got better and better, and there were commercial fluorescent probes for these chromosomes, an expanding range of them, including, as you say, chromosome 21. We began to use it to diagnose these chromosomal errors that would cause miscarriage, or could, in rare cases, give rise to abnormalities like Edwards syndrome or Pateau syndrome, which really develop into highly abnormal fetuses that rarely get to full term. And so we began to use the approach of PGD, taking cells from each embryo and so on for those purposes. And then about the mid-60s, I remember Luca Giannaroli coming up to me at a meeting. Luca Giannaroli, of course, who subsequently became president of the European Society, who works in Bologna in Italy, and saying, what do you think about the possibility of us using this for embryo selection to improve the outcomes in IVF generally? And so for many years, FISH was used with 7, 9 probes, but there was a limitation to this. I know that you, for example, worked very hard to try and do repeated hybridizations and so on, but it was very difficult. And it was only in as recently as 2008 that we actually had molecular tools, as it were, to look at copy number of all 23 pairs of chromosomes, or 24 different chromosomes with the X and the Y chromosome. And I think this has been highly controversial, and but what is clear, I think, from the data is that it confirms the early observations using FISH, that there are a lot of abnormalities in early embryos. And we're still working out the clinical significance of all this. But it's quite clear that aneuploid embryos, that is to say, embryos that don't have a normal number of chromosomes, either fail to implant, so the IVF cycle fails, or it results in miscarriage. And only in rare cases, actually, is it viable enough, if there's an abnormality in the copy number of one of the small chromosomes, will it get through to full term. So now we do see an increasing usage of these platforms that allow us to look at copy number for all 24 chromosomes as an adjunct to embryo selection. And the great thing about that is that it improves the success rate per transfer. So that couples, particularly if they're in the older age group, because many of these abnormalities, particularly those that occur in eggs, increase very exponentially with women beyond the age of 35. And by the time they reach the mid-40s, almost all of their eggs and embryos are affected. And so it can have a very big impact and reduce the number of transfers with these abnormal embryos.
So nail your colours to the mast then. A lot of people say it doesn't work, including the HFEA awarding red lights to it willy-nilly. So what's your view? Does it work?
Well, it certainly works in our experience. Mm-hmm. But I think, you know, that the terms of reference have switched. So that over that 10-year period that I've talked about it being very controversial, when we moved from fish to earlier molecular methods to the latest molecular methods, what has happened is that we've now understood that we can freeze embryos that are biopsied at the blastocyst stage with a very high degree of efficiency. So that now we've moved on to assessing techniques for embryo selection based on the possibility that we might be eliminating from the cohort from each cycle some viable embryos. But, you know, embryo selection techniques, whether it be what we now call preimplantation genetic testing for aneuploidy or any other embryo selection, whether it's just morphology, is only embryo selection. It doesn't change the embryo. And so cumulative pregnancy rates, which is what people focus on now, are not affected in any way. All it can achieve is to try and make sure that a viable embryo is put back. And that has many clinical benefits, which I think are poorly understood or only slowly being understood, particularly in the UK. And so I'm very disappointed, as I know you are and many others are, that the HFEA has not recognized recent work done in this area.
Yeah, very much so. And, you know, it always strikes me as a very political argument more than anything else. Right. So let's move on then. So some would say that, you know, we had those initial headline discoveries. We had, you know, the first single gene disorder, the first dominant disorder, the first chromosome disorder, but there was an awful lot of more of the same for 15 to 20 years. Is that fair comment? Let's say pre-2010, just for the sake of argument. Is that fair comment? And were there some major discoveries between those initial heady days and about 2010?
Well, I think you're right. I mean, what characterized that period of time? Again, you need to bear in mind that this was all being done in private labs, and for each patient and each disease, methods had to be developed that were specific to the patients and the disease, and this took a lot of time. And so as a result, what groups did to share this information was to publish a paper on a particular disease. And so, as you can imagine, there were many papers over that period of time that reported specific protocols and hopefully some pregnancy success. So cystic fibrosis in 1992 was the first one. And that was the collaboration we had with Martin Hughes' group that was published in New England Journal of Medicine, because it was the very first specific diagnosis. But then subsequently, there were many, probably hundreds of papers in different conditions. But I would say for me, the standout was really the publication in 2001 by Yuri Valinsky's group. Now, Yuri, of course, was indeed one of the pioneers, and we were aware of his work. In fact, I met him in the late 1980s in Chicago. So he was based at the Royal Masonic Hospital in Chicago in the United States. And he had a group that was pioneering PGD. In fact, they were involved and had their first abstract in 1985. So that was before I became involved. But we beat them to it, if you like, because they, if you like, betted on analyzing the genetic material in polar bodies. And based on my background experience in mouse embryology, I worked on cleavage stages. But they announced that what they'd been able to do, really, which was a tour de force at the time, was to tissue type each of the individual embryos in families with conditions like beta thalassemia, for example, conditions that affect the blood cells. And in this particular case, it was a case of Fanconi anemia, in which there was an existing sick child, but there were no other close relatives that matched that sick child. So there wasn't a possibility then of taking bone marrow from any of those close relatives in order to provide stem cells, if you like, for the sick child, which was the established therapy at that time. So several groups, including Mark Hughes, thought about doing this, but Yuri Valinsky's group was the first to achieve it. And they were able to tissue type the embryos as well as test for Fanconi's anemia, which was a huge technical achievement. And indeed, they had a pregnancy, the baby was born, and the cord blood was isolated at birth. And there are, in many cases, sufficient stem cells in that small volume of cord blood that can be recovered at the birth of the child, which would otherwise be discarded, that could be used for transplantation. And that child was cured of Fanconi's anemia as a result.
So-called save your siblings.
Save your siblings. And now I'm pleased to say that some groups continue this work. And there have been, I don't know the exact number, but there are hundreds of children have now been treated following that kind of approach.
So around the 1990s, a pre-implantation genetic diagnosis society was formed with meetings. Well, it's every year now, isn't it? But at the time it was every 2 years. What are your memories of those meetings, and do you have a favorite?
Well, I have to say the very first meeting was memorable. That was a meeting in Chicago. Yuri Valinsky organized it, and it was in the iconic Drake Hotel, which is a very old hotel in Chicago. And it was highly influential because, of course, I was there to report our early success in PGD, and I actually showed a video of the embryo biopsy. And subsequently, I remember standing in the lobby because they were running the biopsy video on a loop. And Jack Cohen, a very famous, actually he's Dutch, but has always worked in the United States, but actually in the early days worked with Bob Edwards at Bourne Hall in Cambridge, rushed up to me and basically said, I can't believe you put such a large hole in the zona, for all, of course, the reasons that I was talking about. And what we did at that time to make an opening in the zona was to, if you like, blow acidified medium at the zona to dissolve it. So that's acid at pH 2.5. But of course, you have to remember, I was working in medium that was supplemented with maternal serum. So the very thin acid medium floated to the top of the drop and dissipated. And the very high level of plasma in the drop with the medium, of course, buffered any changes in pH. But I think that, so it was influential because he went on to do his work on assisted hatching, because he put it together with a very high level of implantation and pregnancy rate that we reported. And he said, is it possible that the zona is trapping or imprisoning the embryos so that perhaps it's hardening in culture? So that was one of the things. And of course, it was at that meeting that I met Mark Hughes, and we began collaborating to do the first PGD in cystic fibrosis.
Well, I guess part of me was hoping you'd quite say you particularly liked the 13th meeting, because there we all are I feel like I'm keeping you and Robert apart there somehow, but just outside Darwin College at the here at the University of Kent. And there, of course, is Eleanor, looking no older than she did in 1990, looking radiant there. But I particularly enjoyed running that, and it was lovely to get the team back together again.
It was actually a wonderful meeting, and I'm glad you reminded me of that. And that was also influential because I remember this was the early days of working with the American sequencing company, Illumina, and we had a meeting within the international meeting in Canterbury, which actually discussed how we would run a randomized trial to look at aneuploidy for all 24 chromosomes. And it was the first time that we discussed the phenomenon, of chromosomal mosaicism. Sadly, I don't think we realized what the impact that might have on the trial, but it certainly was a very memorable and a very enjoyable meeting, I must say. It was very nice to be reunited with Robert.
Yeah, very much so. No, I did very much enjoy it. It went like a breeze. Turning now our attention to carrier mapping, something that you came up with and invented, and I think I impeded a little bit. Can you give us in 30 seconds a layman's guide to what carrier mapping is?
Well, thank you. I mean, I think it was lucky that we had very early access to a new enzyme, DNA polymerase, that allows you to do isothermal amplification. Mm-hmm. So we got access before anyone else, before any other research group, I think, in the UK at least, in the early 2000s. And we're able to demonstrate that from a single cell, we could now amplify, not using PCR, but just by heating the sample to about 37 degrees with this enzyme in the right buffer, we could get microgram quantities of DNA. And that opened up the possibility of using all kinds of what we call genomics tools to look more broadly. So instead of PCR, which just looks at a single sequence, but to look broadly across the whole genome and actually look at DNA markers, if you like, on all chromosomes. And so we started working with what sound very primitive and limited at that time, Snip Arrays. So what are SNPs? So it's actually SNP for single nucleotide polymorphism. These occur all over the genome, really, on average, just every few thousand bases. And they turn out to be very useful. So some of these at one end are mutations because they're encoding regions, these SNPs. But if they're not encoding regions, they're generally benign, but you can use them as markers. So trying to keep this brief, you look at the parents, these SNP markers in the parents, using these microarrays, so you bleed the parents, get their DNA. If possible, you look at a close relative, and generally speaking, it's an affected child. And in that way, you can interpret the markers within that family so that you can now amplify, using this isothermal approach, enough DNA to put on your microarray to genotype hundreds of thousands of these SNP markers, which are across each chromosome. So this provides you actually with a universal test. So wherever what we used to call single gene defect, i.e. a genetic defect from a single gene that causes the disease, wherever it is, if you know where it is, and of course following the sequencing of the human genome, again around that time, much more information became available and so on, so that you can then just look and see which of the parental chromosomes at that position have been inherited by each of the embryo. But of course, at the same time, you can see if you've inherited one chromosome from your father and one chromosome from your mother. And if you haven't, if a chromosome's missing, or if you have two distinct chromosomes from your other parent, then you know that that embryo is aneuploid. So it's a powerful technique that combines information on chromosomes with a universal, what we call linkage-based test. You're not looking at the mutation, but a linkage-based test for monogenic or single gene defects.
Yeah, I think the thing I like it about it particularly is it just detects that inheritance of whole blocks of chromosome. So you've sometimes described it as sort of individual parental chromosome painting, I think. And you just get that whole block, and then you see the crossover that's occurred during meiosis. And because you're looking at chromosomes anyway, you can spot whether they're extra or missing. So I love it. Obviously, it's quite genius. Now, my next
question. You can actually visualize Mendel's genetics.
Right? Exactly. Well, we always have an argument about this. I always say it's Morgan's genetics, not Mendel's genetics.
It is also Morgan's genetics.
Now, my next question was, has it caught on? And I'm going to quiz you here. Do you know how many carrier mapping cases have been done in the world?
Sadly, I don't. But I do know that it is used by all of the major labs in the US, and they run many cases. And it's actually spread around the world now. There are labs using carrier mapping all around the world. And indeed, you know, the most flattering aspect of this is that individual labs have developed their own approach to haplotyping, or carrier mapping, as we call it. And so it's being used very widely, and thousands of babies have been born. Certainly thousands of cycles.
I can tell you it's just approaching 10,000 cycles as we speak.
I'm glad to hear it.
Fantastic. Right. So any other major breakthroughs you'd point to in the world of PGT in the last 10 years?
Well, of course, one that's been much talked about is the use of preimplantation genetic testing to do polygenic risk scoring. So everything we've talked about up to now involves diagnosing the presence of a mutation that causes a serious genetic disease in a particular family. And I think most people agree that that is worthwhile in helping the couple to avoid those children. But recently it's become possible. So with our increasing knowledge of genetics, we know that many common diseases like diabetes and heart disease have a genetic component. And we know more about those genetic components. And if there's a high incidence of things like type 2 diabetes in the family, you can look now at a range of markers from across the genome, either in patients or adults. But now there's a group in the United States headed up by Nathan Treff, working in New Jersey, that has been able to actually work out an accurate methodology for doing this in embryos. Now, I think it remains to be seen whether couples will want to do this, but I think we should bear in mind that there are some couples in which this almost becomes as much of a risk as a Mendelian condition. And so this is new information, and we're still absorbing this, but whether in fact IVF and preimplantation testing will become more widespread because we can do polygenic risk scoring, I think is an open issue. The other very exciting development, of course, is non-invasive testing, because we've discovered that there is DNA from the embryo in the culture medium, and that we might be able to use this at least to look at chromosomal errors. But that's perhaps for another discussion on another day.
So the study you're most proud of, the one that you wish you'd done, and your biggest regret?
Well, those are very interesting questions. Nothing like putting me on the spot. I mean, I think the most significant thing actually I would probably have done, and indeed it was recognized at the time, and we did publish back to back with Martin Evans, was the study that I alluded to at the very beginning, which is to demonstrate germline transmission of genetically modified embryonic stem cells. Because between 1987 and now, of course, there have been many, many studies that have helped elucidate the function of genes and made it possible to understand what they do and how they are involved in disease. So I think on a scientific basis, that's certainly true. But I I'm very proud of recent work I did, which actually showed that when human embryos that are normally fertilized divide abnormally in their first division, as some of them do, and you can see this using time-lapse incubators when you watch the video. When they divide into 3 cells instead of 2 cells, they actually continue to divide, but never reach the blastocyst stage. And the reason is you've got clones of cells in which 2 sets of chromosomes are dividing to each of 3 cells. And so those chromosomes are dispersed. There are a lot of, so there are missing chromosomes in all of the cells of the embryo, and that seems to block their further development. So I used carrier mapping, not for inherited disease or preimplantation testing, but actually the pattern of recombination as a fingerprint for those individual chromosomes. And that was a very satisfying study for me, and very exciting to see that. Regrets, well, I did.
I've had a few.
Yes. I think the only possible regret, and I don't particularly regret it now, but that in 1980, I did a postdoc at Johns Hopkins. But before I left, I was working with Matthew Kaufman, who became professor of anatomy in Edinburgh. And we noticed when we were looking at pathogenetically activated blastocysts, that is, there was no fertilization. And what we did actually was to take eggs, treat them briefly with ethanol, a low concentration of ethanol that would activate their development. And we could get these through to blastocyst stage and then transfer them and delay implantation. So we were looking at these delayed blastocysts, which had formed a very large inner cell mass. And I put these into culture and let them outgrow, because the outer cells grow out on the plastic. And I noticed cells in the inner cell mass that looked very much like embryonic stem cells. Matt Kaufman was very excited about this, because if they were truly embryonic stem cells, there was the possibility that we would have haploid embryonic stem cells, and that might be very valuable for cancer studies and other things. And so I went over and very nervously spoke to someone I'd not met before, and that was Martin Evans in the genetics department. It's a great thing about Cambridge, with the departments very close on the same. And we got together, and of course he was an expert in stem cells derived from carcinoma cells. And so we made the first attempts to isolate embryonic stem cells. And then while I was away, they succeeded and published the first publication in Nature, which doesn't have my name on it. So that's a possible regret. But I came back to work with Martin Evans. We did indeed isolate the first cell lines in his lab with pathogenetically activated embryos. And so things that let me think. So I think, what was the final part of your question?
I think that was it. No, I was
what I'd like to have done.
What would you like to have done? Yes, of course, I forgot that one.
Well, I now actually in semi-retirement, as you say, just enjoy other people's science.
Right.
So I don't have to do the work anymore, hands-on. I just enjoy other people's science.
Right. So yeah, what do you do in semi-retire? How do you spend your day?
Well, it sounds very restful, doesn't it? I must say. My experience is certainly not like that. I'm still actively involved in research, analyzing clinical data, writing papers. I'm indeed writing up my memoirs.
Oh.
I've set up my own website for preimplantation genetics, and people can access it through www.preimplantationgenetics.com. And various consultancies. So I'm very lucky, I think, to have had a very varied career and still to be involved and so on. And I'm still very motivated to try and move things forward.
Good, good. What's your opinion of germline editing of pre-implantation embryos?
Well, I think it's very exciting, and I'm sure that, well, even in the time that we've been discussing it over the last 2 or 3 or 4 years, there have been technical developments that make it more accurate and so on. But I think there's still a chance that you can get non-specific targeting of the genome. So this is a technique in which you can edit very precisely individual genes. And so the exciting possibility is that we could cure inherited disease. But I think that's a very tough call in an embryo that's so chromosomally abnormal that I think it's practically impossible. And if you weigh that against the fact that we still today, there have been recent publications attempting to do chromosome engineering with this technology that show that there are other effects elsewhere in the genome. And so I think it's quite dangerous to think about this in clinical application. And after all, for inherited disease anyway, almost universally, the couples we work with, half of their gametes do not have the mutation or the abnormality, and the other half do. So it's really much more straightforward and safer, I believe, simply to put the right, to select embryos that have got the right combination of the egg and the sperm cell.
Now, you've mentioned a lot of interesting people throughout the course of this interview. Robert, Mark Hughes, Martin Evans, Yuri Valinsky. Anybody that you've not mentioned that you feel is particularly interesting and worth a shout?
Well, that's very invidious, isn't it?
You don't have to answer. You can say no. That's it.
Well, one of the pleasures of working with assisted reproduction is that there are large international meetings. I think over the course of the last 20 or 30 years, I've made many friends around the world. So it's been a wonderful experience. Clinicians, scientists, embryologists, and so on working in this area. And the only one that comes to mind has been mentioned already. My original mentor was Robert Edwards, who very late in life was awarded the Nobel Prize. He also was given the Lasker Prize, and very late on was awarded a knighthood. He was very charismatic, sometimes difficult, sometimes provocative, but he had a voracious appetite for science, which I think he passed on to all of us. And I think one of his legacies that I think we forget are his journals that he founded. Human Reproduction, Human Reproduction Update, Reproductive Biomedicine Online. And I think that's a wonderful legacy. And so he would be certainly one that I would remember.
Fantastic. So on that note then, what's next for reproductive medicine, do you think?
Well, I don't know. I'm pretty conservative. Many people might say CRISPR, gene editing, or polygenic risk scoring, and so on, or artificial gametes. I do wonder. I think the science. So people have discovered that there are stem cells in the ovary, for example, which is very exciting. But I wonder how long it's going to be before we really feel that it's safe and justified to treat people with artificial gametes. I would much prefer to hope that we could perhaps non-invasively test gametes so that we could put the right gametes together. If we had some kind of imaging technique that we could use that would not destroy the gametes in the process, of course, and we could put those together, I think that would be a huge benefit, because then all of the embryos we would generate. And my current interest, and I don't know how far we'll get with it, of course, in my lifetime, because it's really to understand why, particularly in assisted reproduction, which I've been mainly involved in, why only about half normally fertilized embryos reach the blastocyst stage. So I've mentioned one mechanism, which I was very excited to uncover, but that only happens in a fraction of embryos. And so we need to know more about that. And if possible, treat embryos, change the culture conditions to reduce these abnormal divisions and improve the percentage of embryos that can get through to embryos at these later stages, because we know then, particularly if they don't have any chromosome abnormalities inheriting the egg, that they have a very high chance of implanting.
Well, look, we're rapidly running out of time now, so I'm just going to ask you for last question now and put you on the spot. So in one line, if you were to write a line of your own obituary, what would it be?
Never happier than when he had his head in a large, complex spreadsheet.
Well, Alan, it's been an absolute delight. Can I just say on a personal note, what a wonderful 30 years it has been working with you, because it is also 30 years since we met. You've driven me insane on times, I have to say, but my career has been in far better place as a result, and much, much enriched by it. I can't even say the word now. So everyone, that just leaves me to thank Alan for a fantastic interview, for sharing all these wonderful thoughts with you. I hope he will put this on his website. We'll try and disseminate this interview far and wide. What's next for the next 30 years of reproductive medicine, pre-implantation testing, and indeed for us, who knows? We'll wait and see. Thank you very much, Alan. Goodbye.
Thank you.